Hello, everyone, and welcome to today's webinar, An Approach to Ultrasensitive Detection and Quantification of Copy Number Variations, CNVs, and Mutations. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by NuProbe. To learn more, visit nuprobe.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speakers, Dr. Nicolas Garreau de Loubresse, Director of Business Development at NuProbe, and Dr. Jinny Zhang, Associate Director of Innovation at NuProbe. Dr. Garreau de Loubresse, Dr. Zhang, you may now begin your presentation.
Thank you for the introduction. Hello everyone, welcome and thank you for joining NuProbe's webinar, Ultrasensitive Detection and Quantification of Copy Number Variations and Mutations. So before we get started, just a reminder that the following products and services are for research use only and not for diagnostic procedures. So today's learning objectives include identifying the main challenges of current methods for sensitive CNV detection, reviewing how NuProbe's QASeq technology can increase sensitivity of both CNV and mutation detections from tissue and liquid biopsy samples, demonstrating the development and validation of a custom QASeq panel for the simultaneous and ultrasensitive detection of CNVs and mutations in the ERBB2 genes, as well as other genes, and providing an overview of custom assay development and validation services for biopharma and research applications. Here's an outline of today's presentation. Please note that we'll not be taking audio questions during the webinar. If you'd like to submit a question, do so through the Q&A or chat feature, and please feel free to submit questions at any time, and questions will be answered during the Q&A portion after the webinar. I will now introduce NuProbe. So NuProbe is a genomics and molecular diagnostics company developing novel research and diagnostic tools to improve the detection of nucleic acid biomarkers in cancer from tissue and liquid biopsy samples. Building on a portfolio of technologies including proprietary PCR chemistry, NGS library preparation, primer design, and data analysis software, NuProbe provides a range of solutions to biopharma companies and diagnostic companies through assay development, sample testing, and kit manufacturing services. Here is a selection of recent partnerships between NuProbe and leading companies in life sciences and diagnostics, using NuProbe's technologies across different platforms, including next-generation sequencing, PCR, nanopore sequencing, and different applications and disease areas. I will now let Jinny introduce the QASeq technology developed by NuProbe.
Good morning, everyone. I'm Jinny Zhang, currently working at NuProbe as the Associate Director of Innovation. It is my pleasure to introduce our QASeq technology to you guys. First, let's start with some general background in DNA in oncology. DNA biomarkers are well known for its clinical relevance to cancer diagnosis and treatment monitoring. Among those DNA variants, there are three different categories: single nucleotide variants, short insertion and deletions, and structural variations. In this webinar, we'll be focusing on introducing our QASeq technology that is targeting at accurate CNV detections. Why CNV is so important? From this figure, we can see a broad existence of CNV among all these cancer types, and a high alteration frequency in some specific cancer types like prostate cancer and breast cancer. Detecting these CNV biomarkers can contribute greatly to prognosis and therapeutics. Therefore, we would like to proudly introduce our QASeq technology, which is a highly multiplexed PCR-based targeted enrichment NGS panel that can offer accurate CNV detection and quantitation on a molecular level on multiple CNV genes simultaneously. CNV's importance is getting more and more attention in this industry, and people have developed multiple technologies to quantify popular CNV genes. So what is the uniqueness of our QASeq technology? First of all, NGS panels comparing to ddPCR, FISH, or immunohistochemistry has its obvious advantages. Theoretically speaking, NGS panels do not have the limitation on multiplexing like ddPCR and FISH because they are limited in the selection of fluorescence channels. Another advantage is that NGS can detect mutations and CNVs simultaneously, which can offer more genetic information when patient sample is limited or hard to obtain in large amount. Also, there is a rising attention of cell-free DNA in oncology for early cancer screening, non-invasive neoadjuvant therapy selection, and post-treatment monitoring, where FISH and IHC is not applicable and ddPCR are not sufficient for screening purposes. Despite of all the advantages mentioned above about NGS panels, current NGS panels on the market are difficult to detect sensitively using high-throughput sequencing, with a limit of only 25% heterozygous single copy variation. There are two reasons that mainly contributing to the bottleneck of NGS panels. First is the exponential increased difficulty in highly multiplex PCR primer designs. Second is a low utility rate of the input DNA samples. I will dive into these 2 points in the next several slides. First, we developed a stochastic stepwise PCR primer design algorithm that can design highly specific primers while keeping the orthogonality of primers to reduce dimer formation on each one of the PCR steps. In the middle figure, we evaluated primer designs using an internal bandwidth function, and 3 sets are picked to demonstrate the algorithm performance. In this 100-plex primer designs of primer design set number 1, 2, and 3, we can see that the more optimization involved, the lower bandwidth score will lead to a higher on-target rate. With this algorithm, we increased the on-target rate from 10% to 70%, which will benefit in both sequencing cost and even higher multiplex NGS panel designs. The second innovation of QASeq is our high-efficiency molecule tagging technology. In the left side figure, you can find that the average conversion yield per target in our panel is around 60%, which is 3 times higher comparing to traditional NGS panels that are using ligation-based method. The high conversion yield is beneficial in 2 points. We can achieve much higher sensitivity to 5% heterozygous single copy variation, or we can either use less input amount than other panels, for example, as low as 5 nanogram input, but achieving the same equivalent performance in QASeq in CNV detection accuracy. On the right figure, we can see that the ploidy Poisson distribution is much wider when considering only 20% conversion yield of the input samples. Higher conversion yield can concentrate the Poisson distribution to the observed ploidy, and the concentration of the randomness can be statistically enhanced by involving as many amplicons as you want. When combining these 2 novel technologies in QASeq, we are able to better balance out the raised randomness caused by ligation or PCR amplification, accomplishing the limit of detection of 5% heterozygous single copy variation. We currently have an internal panel built around breast cancer, which contains 226 amplicons in total, targeting at 18 CNV genes and 84 hotspots in 14 genes. We first performed a series of experiments using synthetic reference materials to determine the analytical limit of detection for CNVs and mutations. We can see that in the left figure, there is a distinctive difference between 2.00 ploidy normal sample and the 2.05 ploidy sample with a spike in. In the right figure, we have successfully detected mutations and indels down to 0.2% VF using Horizon cell-free DNA sample standard. We also compared our panel performance with other CNV detection methods, the IHC and ddPCR. We cross-compared 18 fresh frozen breast cancer samples on the ERBB2 gene for all three methods. On the left figure is a comparison of IHC and QASeq test results. We are highly aligned with IHC, except for 1 out of 18 samples that IHC defined as positive, while QASeq defined as true ploidy. The same outlier showed the same ploidy when tested in ddPCR, as the red dot showed in the right figure at the bottom left. Our results are consistent with ddPCR on this sample, showing true ploidy in both methods. We suspect that there is no DNA alteration with this specific sample on the DNA level, but there may be a downstream irregular translation or protein synthesis that actually gives the positive signal in IHC. In the right figure, the vertical blue bar represents the LOD of QASeq, and the horizontal gray bar is for the limited detection for ddPCR. As the comparison between ddPCR and IHC, we can see that there are noticeable IHC negative samples that are called positive gain by ddPCR, while there is none in QASeq. Therefore, we demonstrated a much better concordance with IHC compared to ddPCR. We tested our panel on tumor and healthy fresh frozen samples. The comparison between healthy and patient samples is very distinctive. All healthy patient samples are very stable and close to ploidy. We also get 7 CNV gains and 2 CNV loss costs on the HER2 gene within these patient samples. The right figure summarizes the mutation costs that we got from patient samples simultaneously. We can see that the QASeq can capture mutations down to 0.2% VF with as little as 5 to 8 nanogram input. As a summary of all fresh frozen samples tested, we found 44% of the samples showed a lower level of CNV, and that is the only captured by QASeq, showed in green. This result demonstrated our capabilities in earlier CNV detections that are missing by other methods. Finally, here are some results obtained from the additional clinical samples from the collaborators at MD Anderson Cancer Center, Yale School of Medicine, and Mayo Clinic. In this experiment, we used DNA input between 4.4 nanogram and 8.3 nanogram. From the same exact FFPE tissue section, we can detect both CNVs as well as mutations. We detect many CNV gains and losses, including low-level CNVs, which is visible here in light blue and light red. We were also able to apply the same exact panel to cell-free DNA, reporting both CNV and mutations of the same samples. Note that this FFPE and cell-free DNA samples are now matched samples from the same patient. This result demonstrates that our QASeq pipeline is highly adaptive to different sample types. Thank you all for your attention on the QASeq technology. I will pass this back to Nicolas, and he can provide more information on biopharma services.
Thank you, Jinny, for the overview of the QASeq technology. So I'll now present services that NuProbe offers to access the QASeq technology and support translational research programs. So building on key features of the QASeq platform, we launch an array of services to develop and validate custom QASeq panels. So our goal is to support translational research and early clinical development programs with a unique solution to detect low-level copy number variations such as amplifications and deletions, together with mutations from tissue and liquid biopsy samples. Our assay development services leverage the QASeq technology that Jinny just presented and the expertise of the services team to build ultrasensitive QASeq panels in record time. So here's a summary of key features and benefits offered by QASeq panels. So QASeq panels provide ultra-high sensitivity for mutation and especially copy number variations with up to 10-fold improvement over other technologies such as digital PCR. Low input requirements with as low as 5 nanograms of input DNA. A flexible solution to integrate a wide range of biomarkers, not just limited to copy number variations, but also including mutations and indels. And the compatibility with cell-free DNA for non-invasive testing in addition to tissue samples. So it typically takes about 12 weeks from the initial assay specifications to a ready-to-use custom panel. So every project starts with first a project consultation involving our R&D service team to redefine the required panel specifications, including the targets of interest, being a series of hotspots, exons of genes, with the ability to cover the entire coding sequence. A sample type, such as tissue, blood, or cell-free DNA samples, and finally the alterations, being copy number variations, mutations, indels, or just one of them. We then proceed with panel development and validation using proprietary design algorithm, chemistry, and bioinformatics software. And in terms of deliverables, NuProbe provides two options. The first one is the sample testing and analysis services performed in our laboratory in Houston or at a NuProbe certified CRO or CLIA lab. And the second option is the kit manufacturing services to turn the custom QASeq panel into a standalone off-the-shelf NGS kit for use across multiple sites. So here's an overview of the panel development and validation process using QASeq. So it is divided in two modules, panel development and analytical validation. During the first module, our service team designs and optimizes the custom QASeq panel until we reach high on-target rates. Then the panel and the protocol are locked, and we perform a series of analytical validation studies using reference materials from commercial vendors or prepared by NuProbe in order to determine the assay sensitivity and specificity and demonstrate repeatability and reproducibility. Once this process is completed, the assay can be used for sample testing as a service or manufactured into a standalone kit. So for sample testing and analysis, there are two options. The first option is that we provide the testing services using custom QASeq panels developed by NuProbe in our Houston laboratory. So the turnaround time is 1 week from sample delivery to report, and the entire analysis is performed in Houston. The second option, we provide solutions to transfer and validate custom panels to third-party CRO partners certified by NuProbe for clinical trial assay development in the US and in Europe. Alternatively, the NuProbe also offers the kit manufacturing services to produce ready-made QASeq kits for the rapid development across multiple sites. And the NuProbe team has extensive experience in manufacturing NGS and PCR kits, including bioinformatics software development. We offer OEM and white label solutions, as well as packaging, storage, and shipping logistics. And that brings me to the end of the webinar. So here's a summary of today's presentation. So we highlighted the importance of copy number variations in cancer and the benefits of NGS for detecting copy number variations. We introduced the QASeq technology, a new NGS method that has been developed by NuProbe for highly sensitive CNV detection, applicable to both tissue and liquid biopsy samples. Specifically, QASeq incorporates novel approaches to increase the conversion yield, increase multiplexing, and reduce error bars to provide unparalleled CNV detection accuracy. QASeq has been validated on panels designed to detect both CNVs and SNV alterations. And QASeq is available as a custom assay with sample testing services accessible through NuProbe or CRO partner, or as an off-the-shelf kit. Thank you everyone for attending this webinar. Please feel free to ask us questions using the live platform. You can also visit our website and social media accounts to learn more about NuProbe and stay informed. Thank you.
Thank you, Dr. Garreau de Loubresse, Dr. Zhang, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is for Dr. Garreau. How many genes can be covered by a custom QASeq panel?
So typically we can develop and validate custom QASeq panel for the detection of CNVs and hotspot mutations in up to 30 genes within 12 weeks. With a larger panel is feasible, but likely it will take more time to develop.
Thank you. Our next question is again for Dr. Garreau. Could you comment on how custom QASeq panels are transferred to CRO or CLIA laboratories?
So in this case, once the custom QASeq panel is developed and validated, so the assay is transferred to a manufacturing team where research use on the kits is manufactured, and the kits can then be transferred to a CRO CLIA laboratory partner. So we have several partnerships with CROs in the US and Europe, or we can work with your preferred laboratory.
Okay, thank you. Our next question is for Dr. Zhang. How many amplicons are needed to get the limit of detection, and can you do more amplicons to make it even better?
Yeah, so the current panel that we're using is around 50 amplicons for one gene to get the detection limit of 0.5% heterozygous single copy variation. But I mean, theoretically speaking, you can do more amplicon, but the more noise of the PCR and the sequencing errors will not really benefit on introducing more amplicons, because after that, the error will be coming instead of from the panel itself will be something else. So I would say probably 50 will be a good point to start, and if we have better platform, we can do more.
Thank you. Next question is for Dr. Zhang. Does QASeq have any other advantages comparing to other detection methods?
Yeah, so the advantage that we have been emphasizing in this webinar is focusing on the CNV detection cost. So, but we have a more advanced technology with the molecular tagging. So that actually can reduce the background noise, which actually gives you a higher sensitivity. So this same technology is the core also applied to the QASeq that can benefit both on the CNV cost and the variant cost.
Thank you. Our next question is for Dr. Garreau. For the kit manufacturing option, does NuProbe provide the bioinformatic analysis software as part of the kit?
So yeah, for each custom QASeq kit includes a bioinformatic analysis software together with the reagents required for the NGS library preparation. And so the bioinformatics analysis software would take would generate a summary report of key findings in the sample tested using the panel, and the input would be a FASTQ of data generated from the sequencing run.
Thank you. Our next question is a three-part question. Can QASeq be used to detect CNVs between homologous blocks within a human chromosome? What is the highest homology that your panel can detect between? And how does your technology compare to MLPA?
Jinny, would you like to answer this question?
Yeah, so honestly speaking, we didn't actually have the idea about our technology comparison to MLPA. And actually, I got a little bit confused about what do you mean by homology that your panel can detect between. So, do you mind that we answer your question like after this seminar? Like we can do some deeper research on this specific question.
Yes, of course. All of these questions you can direct to the attendee through their email. And this happens often where there are questions that are really complicated and require elaborate responses. And so, yes, you will be able to answer their question via email. Our next question is, are CNVs measured across the whole sample? If yes, then what is the effect of contaminating normal cells on the CNV limit of detection?
CNV, so actually for NGS panels, whenever you do panel, basically we don't pick the sample to be input. So yes, we are measuring across the whole sample. As long as you have the molecule for the targetable CNV, like the primers, they will be detected. And the CNV limit of detection is based on the sample level, not by so it will be impacted by your tumor percentage and how many percentage of the normal cells you can have. So it's kind of the sample quality before you actually input into our panel. So what we actually give out is the actual CNV number we can get from this specific sample. So I think it's not really we consider as contaminating. It will be more like we consider it as normal CNV background.
Thank you. Next question, what are the LODs for CNV and mutation detection for FFPE and cfDNA? Can it report biallelic mutations?
Okay, so the LOD for CNV and mutation for FFPE and cell-free DNA, they are basically the same. So our panel can adaptive to both sample types. It's similar to what I answered for the previous question. So we don't adjust our panel based on what is the sample being inputted. We just report what we saw from this sample. So the LOD will be still the same if you use like a 10 nanogram input of the FFPE or a cell-free DNA sample. We still can detect down to 5% heterozygous single copy variation or 0.2% VF for the mutation detection. For your second question, sorry, I don't have sufficient knowledge to answer that right now, so I can reply to you back after this seminar.
Great. Well, thank you again, Dr. Garreau, Loubresse, Dr. Zhang, for your time today and your important research. We would also like to thank LabRoots and our sponsor, NuProbe, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.